Okay, guys, thank you so much for coming. Today we're going to do review for midterm 1, and it's going to cover all the material we've done. I've kind of condensed it into bits, and I'll kind of show you also, how to kind of answer some of the long answer questions and some of the things that I will be asking of you guys. And so all lectures 1 to 5 will be on here. And I'll first of all just talk about the format of the exam, so you know what to expect. So exam format, we have section 1. By the way, the quiz is now open too on Connect, okay? And it's due on Monday, the day before your your midterm exam. Okay, so do it a few days before the Tuesday, so that you can kind of study off of it. It will have 37 multiple choice questions, which will be similar to the 30 multiple choice questions in section 1 here. Okay, because it will be multiple choice as well. Okay, and you will answer 30 times 1, which is 30 points. Okay, and then section 2 is kind of short answer and definitions, like I can say define osmosis or define diffusion, something like that. And you will have in this section, you will answer 5 questions out of 6, and they'll be worth 2 points each, and that's 10, right? And then section 3 will be long answer. Okay, and this will be you only have to do 4 long answer questions out of 5 times 5 points, which is 20, and then that's 30, 40, 60 points. Okay, so this will be the general format of all the exams. Okay. And in these long answer and short answer questions, I have a bonus part. Like after I've asked you to say list the 5 functions of skin or whatever it is, and then I can say, oh, okay, what are the functions of the epidermis? And then 3 points. So bonus points could be anything from 1 to 3 points, additional to the points that question is worth. If it's a 2-point question and you get a 1-point bonus, instead of getting just the 2 maximum points, you get 1 bonus point. The only thing about the bonus points is that you have to pick that as one of your 5 or one of your 4 to do the bonus part. You can't just pick 4 questions and then do your bonus or on another question, to increase. That's like double dipping. So double dipping is good if it's chocolate, but not here. Okay. So you have to choose it as one of your questions, because there are some 1 or 2 questions that may not have bonus parts on them. Okay, so and the bonus parts is a little additional challenge, so that you kind of have to earn it to get the. You know what I'm saying? So they will be asking why something happens, or things like that, or to explain something for some of the parts. Okay, so that's the exam format, and it's 1 hour 15. It's the length of the period. So you should have plenty of time to do it. Yeah. Let's say maybe 5 of these questions will have a bonus part, and maybe 1 or 2 don't have a bonus part. They will each be worth 5 points. But the question that is a bonus section will be additional 1, additional 2, or 3 points. Okay, and the bonus questions will be sprinkled throughout. It's not going to be like one bonus question that you do at the end or something. No, they will be sprinkled throughout that. And usually I like to place bonus questions where it's a little harder. A question that's a little more challenging, I'll select maybe 3 bonus points on top of it, so that if it's a 5-point, you'll be getting 8 if you get everything right. You know what I'm saying? Just as an additional challenge, so that's how it is. That's why if a question doesn't have a bonus and you don't choose it, you can't then pick a question and just do the bonus part and expect to get the marks from it you want. Okay? Once you pick it, you can do all of the questions or just do the regular question and leave the bonus, because it's up to you how you do it, okay? Okay, so we give you the freedom to choose what you want to do. As long as you pick your 5 questions or your 4 questions in the other section, I'm good with it. Okay? And then once you pick that question, you can just do the bonus if you want by yourself, or you can do the regular question by yourself, or you can do both. Okay, but it has to be one of your 5 or one of your 4. Any more questions about that? I think that's good. So the first lecture was on just the general organization of the body and the studies, and then of course we have anatomy. You know what anatomy is, right? It's the structure. So in the exam, I'll ask you about, characteristics, and I'll ask you about functions. If I'm asking about characteristics, I'm talking about the anatomy part, what you can see. If I'm asking about functions, I'll be physiology part, what it does, how it works, the processes, and so forth. So you have to be careful in the exam, because sometimes I mix and match and say, give one characteristic and give one function. You may give a correct function. If I'm asking for a characteristic, then it won't be correct. Or you may give the correct characteristic, but if I'm asking for a function, then it's not correct. You see what I'm saying? So you have to be careful for that. All right, so that's the definition, of course, the set of structure. All right, and it's split into, like surface anatomy, right? It's split into systemic, looking at the systems, and then it's split into regional. Like you could do the anatomy of the head or the anatomy of the arm, you know what I'm saying? That kind of thing. All right, and then definition for physiology, we said this is function, right? And then of course, this physiology is divided into different organisms, because different organisms have different physiologies. They work differently, and it's influenced by the environment and so on. So like right now we're doing human physiology. You see what I'm saying? It's slightly different from other organisms, because other organisms have different challenges and they have to adapt to different situations. And then we can split it into the levels of organization, right? Like we could do cell physiology, right? Or we could do tissue physiology or organ physiology. You know what I'm saying? That's how you split the physiology part. Okay, so I just want you to understand these studies. Sometimes I can say, okay, we're looking at the structure of the hand in a multiple choice question. We are studying, then I'll say physiology, anatomy, biology, whatever. And then you say anatomy, right? It's a structure. Those kind of things. So that's why we need to know that. And it helps you to, determine which is a characteristic and which is a function, and how to answer your questions in the exam, if you know those studies. So of course you gotta know the 6 levels of organization, okay? Starting with the chemical, where you just have the atoms, and then the molecules, and the make-up organelles, which make up the cells. And then the cells put together with the extracellular matrix make up the tissues, right? Then the tissues put together make up an organ, okay? Then the organs put together make up a system, like the urinary system, which is the ureters, the bladder, the urethra, and the kidneys, right? So in the exam, I usually give you just like a 5-point question, and then I say, okay, list the levels of organization in the human body and describe them. You talk about the description of each of these, like I just said real quick, and then sometimes I say, oh, okay, can you list the 6th one for a bonus point, and then you get 6 points instead of just a 5, right? That's how it works. So most of the questions that lend themselves to the long answer type questions, I'll tell you, but it doesn't mean they're all going to come, because there's only going to be 5 questions. You know, you just have to know that this is a potentially a 5-point question. You know, and the rest of the other material will be in the multiple choice questions. So that's why it's very important to do the quiz before the exam, the midterm, because it will really help you. And this time, the quiz, I have set it up so that it will tell you all the solutions and how to work out and what you got wrong and the correct answer and why the right answer is the right answer. That way, it kind of gives you a system to work out most multiple choice questions. You know, if I give so it will help you in the exam like that. So now, these are the characteristics of life. Again, there's 6 of them. So you got to be able to take me about responsiveness, being able to respond to something, Because if you're not able to respond to stimuli, changes in the environment, you might suffer and you won't be successful. Growth, you got to go in cell number, cell size, extracellular matrix, all that. And then, development, changes over time. So I will be asking you to not only to list them, but to explain them. To what is metabolism? What is metabolism actually? Real quick. Yeah, all the chemical reactions, all the sum of all the reactions that occur in the body, that's metabolism. Okay, and then organization. What about organization? Just like the name says. What do you think organization is? Yeah, in cells. There's a specific function. Everything, like the organs and the organ systems, are given a specific function. And organization is when somebody is assigned to do something, they do it, and then somebody else is assigned to do something, and you're supposed to work together. And there's a certain relationship between all those, or among all those organs. So that's organization. Okay, so explain it like that. And responsiveness, and so of course respond, and then reproduction, you gotta be able to reproduce. Otherwise you become extinct in the environment. Okay, so be able to describe all those. Okay, and then here, of course, homeostasis, you're gonna know that, the concept, because that's what the body does. All that organization is so that the body will do what? Maintain homeostasis, which is maintaining a relatively stable environment, and it doesn't flatline. Relatively stable because it will fluctuate about a set point. Because we talked about it last time, it's way too expensive to maintain it at exactly that level. Because any deviation you have to correct it, any deviation. So that brings us to the components you need, right? So you need something that will be able to detect the environment, like a receptor. You need that, and then you need something that is all the set points. That is all the knowledge about what the value is supposed to be, the control center, the brain. Okay, and then you need something that's the workhorse that's going to do the work to change whatever that variable is, like muscles, glands, and so forth. All those are effectors. All right, and then it brings us to homeostatic mechanisms, right? You gotta know about homeostatic mechanisms. Okay, you need those three components, but a homeostatic mechanism, you start with the receptor that passes on the information through a sensory nerve to the control center. The control center will compare it to the set point, and then it will decide whether an action is necessary. If an action is necessary, that's the only time when an effector will be told to do something, okay? To take it up or down. So that brings us to the negative and positive feedback. I need you to know the definition of negative and positive feedback. What is negative feedback? What happens there in negative feedback? Yeah, it's the opposite. Correct. So it's the opposite of what? Of the change, right? Of the stimulus. Okay. So negative feedback takes the change or takes the variable back towards the set point. Okay. What about positive feedback? Okay. What is it and why is it harmful? Yeah, because it makes it worse, right? If something is added 1, it will add 10 and 5 and 6. That means positive feedback moves away from the set point. No good. That will kill you. In fact, it does, for the most part. So in the exam, I'm asking, why is positive feedback bad? You tell me that, right? Because positive feedback exacerbates the initial change. It makes it worse, which means it will move further and further away from the set point. So most of the mechanisms that control homeostasis are negative feedback. Okay? Because I will ask you to define these things, and I will ask you why positive feedback is bad. Okay? Do that. Alright, so, and then this just shows you the anatomical positions, which is this, really. Be able to describe that. It's the reference point for which everything is in the directional terms. So when you're lying supine, you're lying face up or down. Up, and then front face down. Alright, and then the directional terms, things like anterior, posterior, superior, caudal, to the back, cephalic towards the head, ventral towards the bottom, four-legged, dorsal, four-legged, all those things you have to know those terms because they'll help you to describe even the planes as well, the body planes. Sagittal will divide the body left, right, front, back. Which one? Left, right. Sagittal is left, right. So, and then coronal or the frontal will divide into front, back. All right, and then transverse will divide into superior, inferior. So you need to know those. Okay. So, so that's why the reference point is important. You have to know that some of these things are sprinkled in multiple choice questions. These details. Okay, good. And then of course you have to know the body, the membranes, the serous membranes. So for the thoracic, we've got the pleural, and also the pericardial, because it's within the thorax as well for the heart. And then for the abdominal and the pelvic, we have the peritoneum. Okay, so know about those membranes. Know that there's two types of serous membranes. One is called visceral, and the other is parietal. And the visceral one is in close contact with the organ because it's soft. That's why it's called visceral. Because visceral means soft organs. And then, of course, the parietal is one that's on the outside, kind of demarcating the body cavity. And what's between parietal and visceral serous membranes? The fluid, which does what? Lubrication. So know those functions and that arrangement. But in the small intestine, in the abdominal cavity, because we have a bunch of small intestine, and it's a long, convoluted tube, it needs mesentery to kind of put it into some kind of order. Otherwise, they will be tying knots on themselves, like a hosepipe when it has a kink, it's no good. So you don't want kinks in your small intestine. So the mesentery helps with that organization. So that's the function of mesentery. So know that, and the two membranes, and then move on to lecture 2 now. Okay, so those are the basic things I need you to know for lecture 1. So lecture 2 was the basic chemistry. You need to know what matter and mass is. Matter, anything that takes a space, it occupies, a certain space is matter. And then mass is how much matter is in that substance, right? And then weight would be the gravitational force on that mass, right? Okay, so you know this, and this is basic chemistry. And then of course we talked about physical properties, which is what you can see, and then that will be the anatomy part, and then the chemical properties we said. That's the processes and reactions, so that's the physiology part. All right? Okay, so just keep those in check, okay? Because you want to be able to give a characteristic when it's asked. You want to be able to give a function when it's asked. So you don't flip them like that. Anything you can see with your eyes, the physical characteristics would be, physical attributes would be anatomy, and anything that happens or a reaction or something or interaction between molecules, atoms, and so forth is physiology. So major elements in the body, CHON, which would be carbon and nitrogen, okay? And then the rest of them will be trace elements, or some of them that come in between, which form about 4%. And so the organic compounds, the major organic compounds, stem from these 4, okay? Starting with the, but we have to talk about how those elements join up, right? Before we can form those compounds and those molecules, right? So we have what are called ionic bonds, right? What are ionic bonds? What's happening here? What are happening to the atoms first before you can form an ionic bond? Yeah, an attraction between positive and negative, which means they must have done what? The atoms lost or gained an electron, right? So the positive one is called the cation, and the negative one is called the anion. And then the positive and negative attract. All right. So here we have cation positive, anion negative. Okay. Some of these questions about these bonds, I will sprinkle them in the choice. Okay. And then covalent. What is a covalent bond? Sharing electrons. Okay. And this can be done equally or unequally. If it's unequal sharing, then it's going to be a polar covalent bond. Okay, which brings us to the hydrogen bonds, right? What do you need for hydrogen bonds to form? Which two major elements do you need? Nitrogen or oxygen? Very good. So, because why? Because these are missing 2 electrons in the valence shell, which means they really have a great affinity to pull electrons towards them. So, if you have a small atom like hydrogen, that one lone electron that is surrounding it is going to be pulled way towards the nitrogen or the oxygen. So, therefore, you're going to have a slightly positive charge on the hydrogen, and then a slightly negative charge on the part where the nitrogen or the oxygen is. So that's how you form hydrogen bonds. That attraction between the slightly positive charge and the slightly negative charge is going to be called hydrogen bonds. In the exam, I'll ask you to define hydrogen bonds. Don't say I didn't tell you. Right. Okay. And then, so now we have the reactions. Now we get our molecules. Now we're going to have reactions. So the synthesis reaction would be putting together things, right? Making bigger. So be able to describe that. Making a larger substance from smaller substances or smaller chemicals or molecules, all right? And the synthesis reaction is a dehydration because it will remove, in fact, let me do this. So this is an amino acid, right? And this amino acid 1 here with an R group. Okay, and then we have C, H right there. And then this is the second amino acid. Okay, this is L, and this is H, C, and R group 2. All right, this is the C, and that's an H. And then here you have the C. Oh, there's an H here, okay, and then an H here. So when you're having a dehydration reaction, you're removing water from these 2 molecules, right? Okay, and then so this and that equals the H2O that you remove, and then you have a dipeptide. And then when you go the opposite direction, you add that water back, and then you have, so it's a hydrolysis, you add water and you break for a decomposition reaction. So I want you to be able to explain this and what is the synthesis reaction and what is the decomposition reaction and what happens with the water. How do you use water in both of those? Okay, because that's going to be in the exam. All right, and then yeah, you need to be able to explain the rates of chemical reactions and how a catalyst will catalyze a reaction, right? How does it do it? How does a catalyst speed up the rate of the reaction? Yeah, it lowers the diffusion. Very good. Okay, and then temperature. How does temperature affect the rate of a reaction? Higher temperature equals faster rate of reaction. Okay, why is that? Why is it when there's a higher temperature, the reaction goes faster? I'm moving a lot faster, and the chances of them banging into each other are much greater. That's why the speed limit is 70. It was 100, my God. We'll bang each other all the time, right? So good, because I'm going to ask this question in the exam. I'll list all the factors that affect the rates of reactions and explain how that happens. And then, of course, particle size. We talked about that. Particle size, smaller the particle size, the faster the reaction. One. What's that? Yeah, more surface area is exposed, and also smaller molecules move much faster. They're easier to move than it is cumbersome. So then the chances of them bumping into each other is much greater. Okay, very good. Concentration. How does it affect the rate of reaction? High concentration equals high rate of reaction. Why? The more molecules you have, the greater the chance of them bumping into each other. You know, because it's just way too crowded in there. Because for you to react, you have to come together. Okay? It's just like for 2 people you have said they gotta come together somehow. They can't be having it like remotely. Maybe in the future years we might be doing that. We wear a helmet or something, and then But for now, you have to come together. So that's the same thing with this. The more there is, then of course the greater the chance. So I'm going to ask you to explain this and tell me how they affect rates of reactions. And then of course we move on to the biological organic compounds, right? So all of them are based on those elements we talked about, CHON. So we start with carbohydrates which have CHO, right, in the ratio 1 to 2 to 1. Okay, and so they got water in them, so that's why they got carbohydrates. And they're really used for what? Really? Energy as well. And on the cell membrane, they're used as marker for communication molecules. Remember, we talked about those glycoproteins and those glycolipids, and it's the carbohydrate part of it that sticks outside of the cell membrane, which is what is used in cell recognition, cell communications. Okay, and we'll cover that a little bit when we talk about the transmembrane proteins. Okay, so carbohydrates do that, and then lipids, of course, only CH, very long polar, very little oxygen as well. And then those ones we talked about, how they're saturated and unsaturated, and I'll show you another clip on lipids. So you gotta know the different types as well, like the fats, the eicosanoids, steroids, right? Phospholipids. And phospholipids, in the morning I asked them, where are phospholipids located? What's that? Yeah, the lipid bilayer. And they were like, I was like, guys, you should know that without thinking. By now, you should know that phospholipids are in the membrane, the plasma membrane. So you guys are doing good. Okay? Keep it up. Okay. Don't tell them I said that. So proteins, of course, we got CHON, and then we got S. We're starting to add some of those in the middle, like the 4% of the body, right? And the proteins, they can be structural, and I'll show you a slide of that a little bit. And most of them are enzymes, right? Like we'll talk about that. And then, of course, nucleic acids, these are the genetic material, and there's CHON and P, and phosphorus. Okay, those are the nucleotides, phosphate, a 5-carbon monosaccharide, and then a base, a nitrogenous base, which can be 2 ring or 1 ring. Okay, so we need to know that, and that's genetic material. We'll talk a little bit more about the other 3 compounds. The carbohydrates we won't, but we will talk about them about when we talk about the proteins anyway, because they're important. So the fats you need to know, these are the triglycerides, right? They got glycerol and 3 fatty acid chains, and they could be saturated or unsaturated. The unsaturated have unsaturated, double bonds, and then the saturated ones, those are the bad ones, because they're so unreactive, and they just tend to settle and they will clog up blood vessels. So you need more of the unsaturated, especially the polyunsaturated, which have more than one double bond on the carbon double bonds. And then phospholipids, of course, the membrane. Okay. And you guys were good to know about that. Steroids, what do they do? They are hormones like testosterone and estrogen, okay, and they control reproductive function also. And then eicosanoids, they're sort of hormone-like substances, and these have 20 carbon fatty acids. They are like 20 carbon fatty acids, and then of course fat-soluble vitamins A, D, E, K. All right, all those are fat-soluble vitamins, very important. D for calcium absorption and so forth. All right, and then lipoproteins. What are lipoproteins used for? What are lipoproteins used for in the body? What's the function? Lipo, so it means it's a protein and a lipid together, right? Okay, when we are transporting what kind of substance in the blood? When we have problems, when we are transporting what kind of substance in the blood? Huh? Lipids, right? When we are transporting lipids in blood, we may have a problem. Why? Because blood is an aqueous environment, right? And blood, have you tried to mix oil and water lately? No, they don't mix. Okay, so that's where you have the lipoproteins coming. The lipoproteins kind of give some sort of solubility to lipids, so it's lipid transport. Okay, lipoproteins are for lipid transport. Okay, so because I might ask you to list some fats and some of their functions. Sometimes I do that. Okay, so lipoproteins, transport of fats in blood. Okay. Because the protein gives it some sort of solubility, because it's a protein that is polar. So we talk about proteins now, all right? Because the proteins, they affect chemical reactions, because we said most proteins are what? Enzymes. Yeah, very good. Okay, and then structural, such as what does that spell? Collagen. You know, collagen is a protein. It's a structural protein for tendons and so forth, right? And muscle contraction. We haven't done muscles and yet contraction, but we'll do it soon. Muscle is protein. Okay, and the proteins that slide up against each other when muscles contract, those are proteins. So muscle contraction. That's why proteins are involved in that. And then, of course, characteristics of enzymes. They're going to be specific. They're going to speed up reactions, right? And they're going to be globular. Most of them are globular because the globular ones can kind of mix with most things, and again, it works with particle size. That's why they are globular, because they are more accessible. And then, of course, you can control the rates of reactions, or you can control the enzyme by affecting its concentration, how much of the enzyme there is, or by affecting the rate of the activity of that enzyme, right? So all the body conditions, all the metabolism in the body is controlled by affecting that. The activity of the protein here, which is the enzyme, and also the concentration of it. So if the body wants to have high reactions of a certain reaction, guess what it's going to do? It's going to create more of that protein. It's going to go to the DNA, copy the DNA for that, the gene for that, come in the cytoplasm, form a whole bunch of that protein, and then we have more higher reactions. Okay, so that's how the body controls the metabolism of the rates of reactions. So be able to explain, of course, these characteristics of enzymes and how you can manipulate or how the enzymes are controlled and how they control the body. So nucleic acids form the DNA. Okay, so there's two main nucleic acids, RNA and DNA, right? And we talked about it last time. The bases in DNA: adenine, guanine, cytosine, and thymine. Okay, and we said DNA. What is the monosaccharide in there? Deoxyribose. Okay, it's just ribose without the oxygen. Okay, and that's it. And then, of course, the phosphate and the base is what forms the nucleotide, which is what makes up the nucleic acids. All right. And then DNA, of course, is the helix, the double helix. Okay. And then RNA, again, the bases are adenine, guanine, cytosine, and uracil instead of thymine. That's another difference between DNA and RNA, because sometimes I ask, well, what are the differences between RNA and DNA? And then you talk about how the one is double-stranded and the other is single-stranded, how the other one is deoxyribose for the monosaccharide, and the other one here is, what is the monosaccharide in RNA? Ribose, yeah, ribose. And then, of course, uracil. It's got one of the bases instead of the thymine, and it's single-stranded. Okay, what are the varieties of RNA? We got mRNA, which is messenger RNA, transfer RNA, which transfers the amino acid to the growing protein chain. And then what else we got? rRNA. You guys are good, man. You guys are spitting this out like it's nothing. Very good. Ribosomal RNA, which is attached to the ribosomes. All right. In fact, the ribosomes are one of the non-membrane-bound organelles, which we're going to talk about in a little bit. Okay. And we'll talk about the functions of them. So, lecture 3, organelles, cells, and membrane transport. Good. So, the cell is organized, right? And there's going to be 4 major components of the cell. We know what the 4 major components of the cell, right? We get what? The plasma membrane. We get what else? We get the cytoplasm. Then we get the organelles. Then we get the nucleus, right? The nucleus is an organelle, but we include it separately. Why? Because it has the genetic material in there, right? Okay, so, which instructs everything, you know, about what kind of protein that cell makes, what kind of everything, you know, in the structure of that cell. Okay, so the nucleus is a very important organ. So, and then of course we know about the cytoplasm is made of 2 parts, all right? So, what are the cell functions? You know, communication, okay, and then synthesis, we make molecules, and metabolism, creating energy, right? Okay, and then of course reproduction. You know, this is like cloning, getting cells to replicate cloning, and then this is making the gametes that make a male and female gametes. When they unite, then you form another organism. Okay, so gotta know the cell functions. I may ask a 2-point question. Okay, list the functions of the cell. You describe them, and then okay, another bonus part. Can you list another function of the cell and explain it? Okay, 3 points for a 2-point question. Good deal, right? Yeah. There are some people who end up getting more than 100% in these exams. 1 or 2 people have done it in the past because of the bonus. And it can help you because there will be a day when you have a bump time and you get a low point, and this will help you out, when you average it. Okay? So, what is the function of the membrane? It's the outer boundary, right? So it encloses everything. It's going to be selective in what it lets in and lets out. So it's a selective barrier, all right? And then it's going to also be communication, because on the membrane, that's where you have those carbohydrates I was talking about that stick out, that help with cell-to-cell recognition and cell-to-cell communication, right? And then supports, of course. Everything else is kind of supported right inside the membrane. So, functions of the membrane, know the functions of the membrane, which are more intuitive than really anything else. And of course, the lipid bilayer, the phospholipids that I talked about, phospho. Very fast, and then this is that. And we all know why the orientation is like that. The lipid bilayer with the heads, hydrophilic heads facing outside. Because everything is in an aqueous environment. Outside the cell, we get extracellular fluid. It's aqueous. Inside the cell, we get the cytoplasm, which is cytosol, aqueous. Everything is all water, right? All the reactions in the body happen in water, okay? And then, of course, the cholesterol, which is wedged in between to give it stability. So that gives that fluidity. But this is a 4-ring hydrocarbon ring molecule, those 4 rings we talked about. And then the lipids via long chains of fatty acids. So that kind of gives it that fluidity. So that everything doesn't exactly kind of fit into each other like that. Okay, so membrane proteins. So this is when I was talking about the function of these. So I definitely need you to know this. Sometimes I may ask, okay, what's the important part of the marker molecules? Remember, the marker molecules are the ones I was calling glycoproteins and glycolipids. And so the important marker, the important component of marker molecules is the carbohydrates, right? Because those are the ones that are sticking outside of the cell. The protein is wedged all the way inside of the bilayer, right? And the lipids are all facing inside. So it's the protein, it's the carbohydrate that sticks out. So I can ask that question, you talk about carbohydrates, okay? And then, of course, proteins, functions, of course, we said already, the enzymes are proteins. So rates of reactions and so forth. And communication, of course, because they're involved with those marker molecules, the glycoproteins. Okay, and that's where we start talking about cadherins. Remember the cadherins? What do they attach? These proteins attach cells to other cells. Very good. And then we talk about integrins. Those integrins attach cells to what? To, molecules. Almost tissues, but molecules, They attach them to any molecule, and so forth. So that molecule could be metabolized, or it could do whatever. Okay, so attach cells to molecules, the integrins. And then attachment, of course, which we've just been talking about, like some things like selectins and so forth, they anchor white blood cells in place when they're doing their job, so that the white blood cells are not swept away with the blood. So they anchor right there and do the job right in place. Where that, and then there's some specialized proteins that will only attach to a specific ligand. Okay, that will activate another cascade of reactions. Like the G proteins, they are attached to some enzyme that will form something else, or a second messenger or something like that. And these membranes, of course, span the cell, and they will attach to the receptor on the outside of the membrane, but because the protein goes right through, then it can stimulate other reactions to go on inside of the cell. So that's what the receptors are. So all those are proteins. Just know the functions of the proteins, and know, what way that function is applied, on the membrane. Right? Then, of course, transport, we have those carrier proteins, right? Remember the carrier proteins that will attach to a specific molecule and move it across. More like mediated transport, right? Then we have the channel proteins. Channel proteins have a certain shape, a certain size of that channel, which fits a certain shape, a certain size molecule. That's it. So that's the difference between a carrier protein and a channel protein. A channel protein will just be of a specific size and shape, whereas a carrier will actually attach to a particular, molecule or compound, that is a specific shape, and then it will literally move it, it attaches to it. Okay, so that's again function of proteins transport. Okay, so just be able to explain those functions. And of course, this is movement across the membrane. So what do we have? We've got diffusion, which you know that just movement of molecules from high to low concentration. And then we get osmosis, which is movement of water across from high to low. So if I ask you to define that, which I might just do that, maybe as part of a bonus or something, I don't know. Then you get to say all those three things, okay? And then of course we get mediated transport, where any molecule is helping out with that movement, or it assists with that movement. So all the active transport where you have like the sodium potassium pump, for instance, it's assisting the movement of sodium potassium. So that's mediated transport, right? And then we've got vesicular transport. So you gotta know this. And then we've got two major types: exocytosis, which is simply moving out of the cell, we define this one, or endocytosis, right? Which is moving into the cell. And there's three types of endocytosis, which is, right? Pinocytosis, yes. And this is movement of what? Liquid, yes. And then this one, phagocytosis, which is movement of solid. And then of course we get receptor-mediated. Receptor-mediated endocytosis. This one tells you what it's doing. The receptor is mediating, another protein is mediating it. So what we normally have is a protein on the membrane like that. This is the protein, and then it's attached to something. And then when this gets internalized, it then goes in here, and then you have that. You see what I'm saying? Like that. So that's receptor mediated. It has to attach to a receptor first on the surface of the membrane. Then that part of the membrane is then engulfed. Okay? So just know those different types to be able to explain it. And then of course the cytoplasm is made of two things. The cytosol, which is really the fluid component of the membrane, and it includes the cytoskeleton, the microtubules and so forth that anchor and hold these organelles in place. And then the organelles, which is the other extent of the cytoplasm, right? And then what are some of the membrane-bound organelles? We get the nucleus is one, and the endoplasmic reticulum, the Golgi. You have to know the functions of those two, and we'll talk about that in a little bit. But there are some that are non-membrane bound, such as the ribosomes and the centrioles. What are the ribosomes do? Free ribosomes. What's the function of free ribosomes in the cell? Yes, protein synthesis. But what kind of protein? What happens to that protein? Yeah, it's used inside of the cell. So free ribosomes, if I ask you that question, what is the function of free ribosomes, you say that they synthesize proteins that are used by the cell. Why? Because they are created and then they are already free floating in the cytosol. Okay? Whereas the rough endoplasmic reticulum is a membrane-bound organelle that will then enclose them in a vesicle or a bag, if you will, and then take them somewhere else. So usually, the rough endoplasmic reticulum will create proteins that will be used by other cells, because then they can leave the cell via exocytosis, right? All right, very good. So, because I will ask you about some of those things, to tell me about the functions of these organelles and some of these membrane. So that's good, very good. Excellent. Okay, so we get the rough endoplasmic reticulum, which we said is the main function. It started with ribosomes, because ribosomes do the protein synthesis. So this one synthesizes protein, the rough endoplasmic reticulum. What about the smooth? What does it do? The one that I said is smooth is lipid and carbohydrate synthesis. But then in this one, I'm going to go further to ask you, can you tell me a specific function of this smooth endoplasmic reticulum in certain organs of the body? Okay, such as the small intestine, right? Okay, what does the smooth endoplasmic reticulum do in this form? It is involved in what? The absorption of the fats, right? Remember, it will absorb the fats and then will reassemble them and then connect them to the protein so that they are soluble and put them into blood. Okay, so that's absorption of fats, which is still got something to do with lipid fat synthesis, right? Okay, and then the liver. In the liver, what did the smooth endoplasmic reticulum do? Right? It stores what? With the carbohydrates, right? The glycogen. So the glycogen, it puts it together. You know, the glucose. Once you absorb the glucose, it will be assembled by the smooth endoplasmic reticulum in the liver to make glycogen, which is a polysaccharide of glucose. All right. So that again is got to do with carbohydrate lipid synthesis again. Okay, which is the main function of the smooth endoplasmic reticulum. In the testes, what happens in the testes? The smooth endoplasmic reticulum, what does it do? The testes make what hormone, right? Testosterone. What is testosterone? What kind of compound is testosterone? It's a steroid, which is a lipid. They're lipids, right? It's the type of a lipid. So, that's lipid synthesis again. That's the smooth endoplasmic reticulum in the testes. And then, finally, the cardiac and skeletal muscle. What does the smooth endoplasmic reticulum in muscle cells do? It stores calcium. That's the only one that deviates from the normal function of the smooth endoplasmic reticulum, which is to synthesize the So, in the exam, I'll definitely ask you about the function of the smooth endoplasmic reticulum in certain organs, and you explain to me what it does. And you tell me which organ and determine the function. 4 points, right? So, that will begin. And then we move on to lecture 4. Very good. Tissues. See? So, we kind of condensed it into all this. We've got to go through all 5 lectures. So, the main tissue types in the body, again, of course, is the epithelial, the connective tissue. And I said muscle and nervous tissue, we'll kind of do them later on in later lectures. So this is really going to be about the epithelial and the connective tissue, right? And then where do all these tissues come from? Okay, they come from these germ layers. Okay, about 18 days after conception, you have these 3 layers: the ectoderm, which will give rise to things like the skin and the nervous system, right? The endoderm, which gives rise to the digestive system inside, and then the mesoderm, which gives rise to muscle and bone. Okay, so sometimes I may ask you, okay, what does the endoderm give rise to? What does the mesoderm give rise to? Okay, and from these 3 layers, that's what they call germ layers, because they germinate into all these tissues. Okay, they become all those tissues, right? Skin and bone, skin and nervous system for this one, and then digestive and then muscle. Okay, so I want you to know those main, and then of course for epithelial tissue, trigger happy. Right. Epithelial tissue, the characteristics, right? What are the characteristics of the epithelial tissue? Characteristics. Like these are avascular, right? The epithelial tissue, like the skin, there's no blood vessels. Yeah. Good. Little extracellular matrix. Extracellular matrix. ECM, how about that? That sounds nice. What else? What else do they have? Yeah, and then the one on the bottom is called the basement membrane. So they have a basement membrane. And then the overall function is that they cover tissues. They cover things. That's a function, general function. But there's much more functions. Okay? Because they cover things, they enclose things, they're going to form a barrier. Like skin does, forms a barrier. And it's selective too. It will let certain things in, certain things out. So, and some of these, so here, because I might ask you about these functions and ask you to tell me where you might find that function in the body, right? So, barrier, skin, does that. You give an example of that and talk about it and explain. Then passage of substances, where might you want to pass substances? Like in the lung, we might want to pass through a simple and squamous. Okay, so barrier would be stratified. So those kind of things, okay? Stratification means many layers, so you can protect. And then passage, simple, which means one layer, squamous, which means flat, okay? And that's lungs, okay? Passage of substance and protection, same thing, skin, okay? And then secretion, like the glands, right? So, and then these ones are usually cuboidal, right? You need volume for that, or columnar. You can't have a squamous cell because it doesn't have much volume to be creating anything. Okay? So, and then absorption, of course. Most of them are columnar, right? Like that. Columnar and, you know, some of them have microvilli, especially for absorption. And then some of them have cilia for movement of substances on the surface. Okay? And we'll talk about that. So, I want you to just sort of know these functions of the epithelial tissue and kind of be able to sort of give an example where it might be found, you know, where it's applied. And most of the time, the function will tell you what it is. Like absorption, you think small intestine already. You know what I'm saying? Okay, because that's where you do absorption. Okay, and so forth. And passage of substances within the kidney. Well, kidney to some extent, but it's cuboidal, simple cuboidal, but you're talking lungs. Okay? Right, so epithelial tissues are going to be, because we said there's very little extracellular matrix for epithelial tissue, which means there's more cells. So we're going to classify it according to what there's more of cells. So it's going to be how many cells there are, which means it's layered or simple or one layer, or the cell shape, which means cuboidal or columnar or squamous, flat. Okay, so and then we will, and also the characteristics here lend them to the function. So you will find that simple is mostly associated with passage of substances and things like that, and diffusion, infiltration, and then here pseudostratified, you know, usually because it's a big cell, it's a columnar cell, it's going to be kind of lining these tracts and so forth with cilia on it, like in the respiratory system, bronchioles and whatnot. Okay, and then stratified, of course, it's going to be the skin, and then transitional. Okay, this one it transitions from one cell shape to another cell shape. Okay, and you find it where, like in the bladder, right? Because the bladder will stretch when it's filled with urine, it will stretch, and then the cells, starting from cuboidal, they will end up more flat because they've been stretched and been, you know, more squamous. So that's why it's transitional, right? So and then we all know the shape of this cell, and so the shape lends it to the function, and then so the structural relationship, like I said, with the function. Stratification is a bearing on what kind of function that is, and we've already talked about it. Cell shape is a bearing on what kind of function it is. The cuboidal and more volume the shape of the cell is, the more it lends itself to secretion and production and glands. All right, and then of course free surface characteristics. I need you to know these free surface characteristics, like the free surface in blood vessels, for instance. It's going to be smooth. Okay, so here the blood vessels. Okay, and the free surface with microvilli will be somewhere in the intestines because it increases surface area, small intestine, SI. And then with cilia, where would you find a free surface with cilia? Yeah, the uterine tubes. You know, the uterine tubes as they pass the egg along towards the uterus. Okay, also in the respiratory system, the bronchus. You know, the primary, the trachea, they all have, they are columnar ciliated, so that whenever you trap particles onto the mucus lining them, you know, then it's moved up there, so you can spit it out or whatever, or swallow, whichever you prefer. All right. So, but it moves, you know, mucus and trap particles. So, respiratory system, respiratory system. Okay, and that's that. And then, of course, the cell connections, you know, they affect the tissues. In that tight junctions will form a tight connection between cells to prevent permeability of substances through that. Okay, But gap junctions, on the other hand, they are little protein channels, right, with a hole in it that will allow ions to pass through the membrane when an action potential is traveling through the cells, so that they all contract almost at the same time, like a function of system. So that's very important, like in cardiac muscle. Okay. So the way the cells are connecting to each other, also affects the function of what those cells are doing. Okay. And then we've seen in epithelial tissue there are a bunch of desmosomes and hemidesmosomes, because the desmosomes attach the cells together, especially because it's stratified. There's many cells. So, and then of course the hemidesmosomes bind to the basement membrane. Okay. And so that's how these cellular connections affect the structure and function of the epithelial tissues. See what I'm saying? Okay, so very good. Then we move on to epithelial tissue glands, because the glands are made up of epithelial tissue, and again these glands are classified according to the shape of the gland. Okay, so it could be tubular, it could be acinar, which means it is just sac-like, that it's filled with cells inside, or it could be alveolar, which means it is alveolar. It's sac-like, but it's hollow inside, such as the alveoli of the lungs for passage of gases, right? So those are their structure, and then we have classification according to how the product is secreted. So that we have merocrine that will secrete a fluid, a product with no cell components on it. Then apocrine glands will secrete with portions of the cell with it, right? And then of course the holocrine glands will secrete with the whole cell is secreted out, like the sebaceous glands, and that whole cell is full of the lipids. And then when it bursts, the cellular debris and the lipid inside makes the sebum, which will be like an oily substance that makes the skin soft, right? So that's how you classify them. So just know the structural classification and the secretion time, right? And then characteristics of connective tissue, as opposed to epithelial, we said in this one would be more what? Extracellular matrix. So everything about classification of connective tissue is going to be based on the what there is more of, extracellular matrix. Okay, and then we'll talk about what produces the extracellular matrix and what is inside of it. Okay, so what produces it? The blast. Any cell that ends with the blast, whether it's an osteoblast or fibroblast, produces the matrix. And then the site will maintain the matrix. It's a mature cell that maintains the matrix. Okay, and then the clast will break the matrix, like osteoclast will break that bone so that you can furnish calcium. Okay, so that's what happens. So functions of connective tissue: connecting, structural, and cushioning and support. The brain is protected by the bone. Okay, transport, protection in terms of the immune system, the blood, connective tissue, it's got immune cells in it, right? That. Moving, legs, the muscles are connected to the skeleton, right? Which is the bone, and storing as well, okay? The bone stores calcium and a bunch of phosphorus too, okay? Fat stores energy, loose connective tissue and so forth, all right? Adipose tissue. So, since we got a lot of matrix, we're going to talk about the matrix. What is in the matrix? I'll ask you about what is in the extracellular matrix, and you're going to talk about the ground substance, which is those proteoglycans that can absorb and lose water, so that you can compress and decompress those, like the discs, of course, and some of the other tissues with connective tissue that need to act like a shock absorber. So you get the ground substance in that. And then of course you get the fluid, right? Okay, and then you get the protein fibers. Okay, so you get the protein fibers, whether it's collagen fibers or whatever. So know the components of the ground substance and what they are, right? And then of course I want you to know how the connective tissue is classified and what the function of it is. Like loose connective tissue is like a filler compound. Areolar tissue is going to be found between muscles, Okay, and it's what gives the marbling. You know how they call marbling in beef? It's supposed to make it nice and tender and soft. Marbling is because of the alveolar tissue, which is wedged in between the muscle cells, the proteins. Okay, so that's that dense connective tissue. Where we might find dense connective tissue. Tendons, yeah, of course, tendons, and you know that you find dense connective tissue, and you also find it in skin. You know, in the dermis. You know that reticular dense irregular connective tissue. Okay, so I want you to know this because I might say, okay, can you give me 5 different types of connective tissue and where they might be found in their function? Okay, and then cartilage, of course, you've got the hyaline cartilage, you get the elastic cartilage, you know, and it's found on the fibrocartilage as well. You find it in the joints, between the joints, you find that, or the fibrocartilage between the discs, the vertebral discs, and of course bone. You know what the function of bone is. It stores all these minerals, it supports and does all these things. And of course blood is a fluid matrix which houses all these immune cells, red blood cells, oxygen transport, everything. So you can spot off a lot of things, okay? And you get 5 points, and then I might throw another one. Say, can you give me another connective tissue, get an extra point or something or whatever? There you go. So I don't know. And then of course, this was I said, you just need to know that there's 3 different kinds of muscle tissue. You know, smooth, cardiac, and skeletal. And then that for the nervous system, there's neurons, and then they're supported by neuroglia. And we'll really start this later on. So for now, we'll be focusing mainly on the connective and the, but insofar as, what makes these membranes, this one we've already done. Well, mucus, we kind of talked a little bit about it, and it lines all those systems that open to the outside, like the digestive and the respiratory, all those are mucous membranes, right? And seriously, there's 2 of them, right? But one we didn't cover is the synovial, which is a membrane that houses the fluid that gets joints. Okay, the synovial fluid, and that's the function of that, and that's the connective tissue there. And then inflammation, that's how you heal these tissues, and the symptoms of vascular redness, swelling, heat, it disturbs the function as well of that. So we don't want constant inflammation because the many blood cells in the area, it kind of destabilizes the proper function. So chronic inflammation is no good. All right, and then tissue repair. What happens is, when you're trying to replace the cells that have been damaged, you first have to clot and close the bleeding. Then you inflame by bringing in a whole bunch of blood cells, which will start the production and cleaning up of microorganisms. And then the granulation tissue is what forms the matrix, like fibroblasts. So that's where the fibroblasts come from, the granulation tissue. And remember, we said blasts make up the matrix. And then those cells, after the matrix is made, then it's changed into whatever the cell that needs to be replaced. That's when you do the regeneration. So you have brand new cells to replace the ones that have been damaged. So those are the 4 stages of healing tissues, right? Just know generally just those 4 stages and what's happening in each. Okay, we're kind of doing good because in the morning we kind of got hung up. So this is the last lecture, and we've kind of done this recently, so it's still fresh in our heads. Hopefully, but I want you to know the functions of skin in terms of where they are involved and what tissue it would be. So the protection would be, the dermis, right? I mean the epidermis, right? The layers and the stratification, and then it will protect against physical damage and abrasions. And then what else do we have in the skin? It will protect against UV light, right? And then this will be the melanocytes, right? Because they produce melanin. And then it will protect against what again? What else? Protect against dehydration. Okay? And then there's the lipids in there. The keratin cells, keratinocytes, because they're dry, dead cells, they prevent loss of water, plus the lipids that are surrounding these cells prevent loss of water. What else? Even the sebaceous gland produces an oily substance that goes on top of your skin, prevents water loss. Okay? On top of making your skin soft. Okay, sensation, what are we talking about here? We're talking about things like the Merkel cells. Remember the Merkel cells in the epidermis? What do they do? These are touch receptors, right? So you get touch receptors, pain receptors, nerve endings. Okay, you say all that, that's sensation, right? And then temperature regulation, what we got? We got hair, we got sweat glands, yes, thank you. We got sweat glands. Okay. And then what else we get? Blood vessels, right? Remember the blood vessels? Erythema. When you expose the blood vessels to be more superficial, you lose weight. So blood vessels also help with temperature regulation, and also they help with the well, they don't help with, but they cause color changes, which I'm going to be doing in a little bit, which I'm going to need to know. So they cause color changes. So the skin blood vessels do that. Blood vessels. So there's a lot of things that are involved in temperature control, don't you think? Why is that? Because temperature control is very important, right? Because the rate of reactions are going to be affected by the temperature. All those enzymes that we make in the in the cells, they all operate well at 37 degrees C. So the body goes to all the lengths to maintain the temperature, at 37 degrees C. That's why when you've got a fever, it's trying to kill a bug, trying to make it uncomfortable, and then it's uncomfortable for you too, because you don't operate at those high temperatures. Okay, so there's a lot of things. Even the dermis, because of the thickness of it, covers and insulates, and the hypodermis, which is not even a true layer of skin, got a bunch of fat, it's insulation again, and it reduces heat loss. So everything is to maintain all that metabolic energy that you're producing is used to maintain your body temperature. And then, of course, UV light plus the 7-dehydrocholesterol makes vitamin D. So, that's for calcium absorption. Goes into the bones. And it affects everything, movement, and all the functions of the bones. All right. And then excretion, we said, is a minor, very minor, function of everything the integumentary system, because it's just very little uric acid, urea. But because there are systems that are very well adapted for excretion, okay, not skin. But it does a little bit of it, so we mentioned it. Okay? So if I ever ask about the function of the skin and the integumentary system, we want to talk about that. Okay? And then, of course, I need you to know these layers. Okay? Sometimes I might ask what's the function of the epidermis generally. It's kind of like what? I kind of mentioned it at the beginning. It's mostly protection, right? Because it's layers. What about the functions of the dermis? For instance, because I might say, okay, what are the functions of the two skin layers? And you know that the two skin layers really are going to be the dermis and the epidermis. The subcutaneous tissue is really not a true layer of skin, okay? But we include it because it contributes a lot of good functions towards the function of skin, really especially. Okay, so the dermis does what? Structural strength and flexibility, right? Because of the connective tissue. Remember, the dermis is made up of the reticular connective tissue and the areolar connective tissue, which is in the papillary layer. Okay. And then the reticular layer is 80% of it is the of skin is the dermis anyway, the reticular layer. Okay. So, gotta know the functions of that, and then of course gotta know the layers of the epidermis, right? We're gonna talk about them in a little bit, and you gotta know them in their order too. Okay, like we get the corneum, which is the most superficial, right? We get the lucidum, which sometimes is absent, then the granulosum, and then the spinosum, and then the basal layers on the bottom. Okay. So I need you to know that, and then I need you to know the 4 cell types in here, which I'll cover in a little bit. The 4 cell types in here in the epidermis. Like the melanocytes, the keratinocytes, the Langerhans cells, and the Merkel cells, and what the function of those cells are. Okay, so I need you to know that, which is this. So you need to know these and their functions. So these prevent dehydration, and these UV light, melanin, and these the macrophages, right? the macrophages of the skin of the epidermis, because they migrate to white blood cells when they attack bacteria. And then macro cells, we talked about these attach receptors. All right. So I'll ask about this. Is a 2-point question, and then I'll say throw 2 more of them, and then you get 2 more bonus points, or 1 more, you get a bonus point. Okay. 2 points, and you get 3 or 4 points. And then, of course, these ones I'm going to need you to note them in their order. And then especially that structural and functional and chemical changes that occur from the basal layer going to the most superficial layer. Okay? I need you to know that in there. I'm going to show that in a little bit. So, the structural and functional changes that I'm talking about. And I will be specific about what I'm talking about. I'll say start with the deep layer, okay, going to the most superficial. So you'll be starting with the stratum basal, right, because it's the deepest. And then you go to the spinosum, and then you go to the granulosum, then lucidum, and then you end up at the conium, okay? You go in the opposite direction, because that's how the cells are being made at the basal layer, because it's mitosis, right? And then it's going up. So, what are those structural changes, by the way? We start off here as cube shape, right? Okay? And these are, the function of this layer is to mitosis, right? To furnish a bunch of cells, which will then eventually replace what's on the top. So that's function, and it's cube-shaped cells. Remember, whenever something is producing something, it's got to be cube, it's got to be columnar, because you need the volume for all those organelles required for that. Then in the spinosum, you're starting to produce keratin, right? Keratin. Right? Keratin. Keratin. Okay? And then you're producing also the lamellated bodies full of lipids, right? Okay? The cells are still cuboidal. So those are the cells. And then by the time you get over here, the cells are now diamond shaped, right? Because you're now beginning to flatten them. Okay? So, because I'm going to ask about this, okay, I'm going to ask about the structural and functional changes occurring in the epidermal layers, starting with the deepest layer, which is the basal layer, going to the most superficial, which is the conium. I'm going to ask about it. And then, what is this one making? It's making keratohyaline. Alright? Keratohyaline. Okay? And that's why it's granular in appearance, right? Because the keratohyaline forms granules in it. Okay? And then the lamellated bodies, what's happening to them? They're bursting, and then they're exposing the lipids, right? Okay? And then here, these are really flat cells, and two or three layers, flat cells, and they get dispersed keratohyaline in them, okay? Because this is the layer that sometimes is absent, right? So dispersed keratohyaline, okay? And they're squamous, they're flat. Flat is easier to write. And then these ones now, these ones will be flat, they are full of keratin, and they are flat, and they are surrounded by lipids. Okay, one of the other chemical changes in here is that the cells are coated just under the membrane by a hard protein coat, protein PTN coat, okay, in the stratum granulosum, on top of them being diamond shaped and the lamellated bodies bursting and the cells dying. Okay, so I'm going to definitely ask about this, and then I'll throw in a bonus question asking maybe about that, maybe the function of the epidermis and things like that. Okay, what is the epidermis? 3 points, but that's a hard question. So usually I associate higher bonus points with the harder questions to encourage you to pick those questions. Okay? Because you only get to do the bonus part if you pick it as one of your 5 or one of your 4. Okay, so the bonus is kind of like an added thing, so you kind of have to earn it. So I usually put more bonus points on the harder questions. The easier ones I may not even put a bonus part. Okay, just to encourage you to kind of sort of end the point. And then of course skin color. This one, you know, I need you to know about it and be able to explain it. Okay, there's so many ways you can affect skin color. And I was talking to Jesus a while ago about the keratin, right? And the keratin will cause that if you have too much of it, of course you have the yellow color. But another way you can create yellow color is by alpha keratin. hepatitis, if you have hepatitis, because what does the liver do? It metabolizes pigments. Okay, like bilirubin, and so forth. If they're not metabolized because the liver is failing, then you get the jaundice, you get the yellow pigment in your body. So that's a pathological condition which can affect skin color. Then the other one is erythema, which is like when we say blushing, when you blush, it's pretty much exposure of your blood vessels to the surface, right? And therefore, again, it also happens to be a way to lose heat. Okay? Because when the blood vessels are more superficial, you lose heat. But it also affects the skin color, because when the blood vessels are more superficial, you see the red, especially in fair-skinned individuals, okay? And that's how you affect skin color. But the basic way, or the general way you affect skin color is the melanin produced by the melanocytes. And we said everybody's got 8% melanocytes, but it's the amount of melanin and the size of the melanin granules you produce that affects the skin tone, okay? So that's another way of affecting. And another way is environmental, where you have tanning. Like fair-skinned individuals like to go tanning, or they go on a tanning bed, or they go to the beach or whatever, lay down there in tan. So that's another way, and they become bronze or darker. So that's another way of affecting skin color. So and then cyanosis, oxygen. Oxygen content in blood will affect. If there's less oxygen content in blood, you become blue. Like most mothers know about cyanosis. When the kid is turning blue, they rush them to the hospital because they're not breathing, they're not getting oxygen. Okay. So in the exam, I'm definitely going to ask about how you affect skin color, and you explain it, just like I've been saying. Okay. So just think of carrots. Okay. At least when you're doing that. And then of course, I need you to know how melanin is transferred to the keratinocytes. Okay, you know how the melanocytes are in the basal layer and how they grow those processes that weave through between the cells. Okay, I'm going off, but we'll be done in a little bit. And then they get engulfed, those processes, by the cells, by the keratinocytes, and then that's how you transfer the melanosomes to the other. So know about that. Okay. And then the dermis, of course, we got the two layers. It's a connective tissue, and the epidermis is epithelial tissue, right? Okay, this is connective tissue, and this is loose connective tissue in here, and then this is loose or areolar, and this is reticular, which is dense, irregular connective tissue. Remember I was saying dense connective tissue at that time, and this is 80% of it. So know the functions of it, right? And the characteristics of the dermis is that it's got these two layers, and it's connective tissue. It's made up of connective tissue. It's got a lot of extracellular matrix, of course. And then subcutaneous tissue, it stores fat. It's that layer that we say is not true layer of skin, but because of the contribution of functions to the skin and the integumentary system, we kind of include it. But this is what it does. It stores fat. It's an insulator. It's a shock absorber. Because that subcutaneous fat is very important. It also works with temperature regulation in terms of insulation. All right, and then hair glands and sweat glands. We need to know these insofar as they are concerned with temperature control. And the nails also protect. They're not involved in temperature control, but they protect the tips of your fingers and your toes. All right, then they're filled with hard keratin. And then of course, sweat glands. We know about the two types: the merocrine and the apocrine ones. That will apocrine ones are more in the armpits and the pubic area, and we talked about how they create body odor. And the ones in the pubic area, the sweat glands, create the odor that's associated with sex, right? And nails, of course, it's the stratum corneum. Instead of having the soft keratin, it has hard keratin, so it's very hard. Hair also has got hard keratin in it, and it's the epidermal layer that creates the hair. So they are epithelial cells. Okay, so hair is not connective tissue. It's not connective. It looks like hair might be connected, but it's not. It's epithelial tissue because it arises from the epidermis, which is connective tissue. Which is epithelial tissue. Okay. All right. So very good. So just know about that, and know the sebaceous glands. They produce the sebum, the oil, which makes the skin nice and soft. And then, of course, these cancers, I just need you to know, the cell that's affected, like the basal cell carcinoma, like the name says, basal cells and it's reduced to malignant. Squamous cell, it affects the keratinocytes in the spinosa, and then it metastasizes, but it can be treated. It affects the lower scalp. The melanoma is one that's bad. That's really bad because it's malignant. It's resistant to chemotherapy. It metastasizes quickly, and it affects the melanocytes, which really protect us from UV. Because without that, and then you have all this ABCD rule, whether it's asymmetrical and it's the border of it's got indentations and it's kind of crooked, and then of course the color, which is dark, all right, and the diameter is about 6 millimeters. That depends on the case, right? And it's very bad when it's 4 millimeters deep into the skin. That's sort of like sure death for that because it's very bad. And this one, I just need you to know about, how, the burns are first degree, second, third degree, and just thickness of burn all the way deep to the subcutaneous tissue. And then the rule of nines is just a way of, treating and, and diagnosing burns in terms of the area and how much grafting you may need. Okay, but I just need you to appreciate how the aging is affected, like slow generation of skin, so it becomes dry. The glands produce less. Sebaceous glands no longer producing the oil. Okay, that's why the elderly have skin that is itchy and dry because the sebaceous glands are not working. The melanocytes decrease in activity too, so they get more cancers. The older you get, the more chances are of you getting dying of cancer and stuff like that because the protection is gone. Right, and then of course the loss of elasticity, which wrinkles, so sometimes I ask about how the skin is affected with age and what happens. Okay, so that's it. Thank you guys. We just took a little bit extra, but so I'm sorry about that, but it was worth it. And then we can start at 5, so we can do on time. Is that okay? We get 10-minute break. Is that good? Or do you want me to give you 5 or? Let's just start at 5. Okay. All right, thank you guys. I appreciate it. Thank you.